you, David. Good morning. Welcome. It's great to be here among you today and tell you about some of the work we've been doing to understand the epigenome, how it is dynamic during development, and how it regulates plant growth and development. So Harvey alluded to some of the discoveries that have been made at Whitehead Institute in plant biology, and I wanted to highlight a few more. Not all from Whitehead, but really push home this idea that although it may seem unusual to have researchers studying plants at a biomedical research institute, in fact, for the past 25 years or 30 years, plant biology research has been really integrated into the biomedical research enterprise as a whole. And that's because plants, the organism that we work in, Arabidopsis, which I'll tell you more about, offers some distinct advantage for certain types of experiments. And so what I've listed here are some of the discoveries that were first made in plants, and then the discovery in plants led to discovery in animal systems. So plants and animals share a system of immunity called innate immunity, which is our first line of defense against pathogens. And there's receptors inside your cell that will perceive if they're being attacked by a bacteria, for example. And those receptors, which are called leucine-rich repeat receptors, were actually first identified in plants. In 1993, these genes were discovered. And based on those findings, by virtue of identity between, or homology between the plant proteins and proteins in humans, they were able to discover that in humans, these proteins had a very similar function. And so one of those receptors in humans was actually one of the first genes identified as being involved in Crohn's disease, which is an autoimmune disease with increased inflammation. Some of the other ones I've listed there are discoveries of photoreceptors that control circadian rhythms. So both plants and animals, a lot of our physiology is on a 24-hour cycle, a day-night cycle. And those discoveries were first made in plants, and homologous genes do the same thing in animals, including us. Very fundamental discoveries about how genes are regulated, how the expression of genes is controlled by RNAs were made in plants, and some of those discoveries have been applied as potential therapeutics in animals. And finally, what I'll touch on more throughout my talk is that many of the factors, many of the proteins that are involved in regulating the epigenome were first discovered in genetic screens in plants. And many of these fundamental regulators of the epigenome are actually altered in cancer cells. Okay, so the other reason we study plants, not only are they great in terms of their use in biomedical research, but of course we all rely on plants for a very fundamental need, for human nutrition. And there is a looming potential crisis on the horizon where over time, so this graph shows yield of various important crops per acre from 1970 projected out to 2050. And so there have been increases in yields in all of these crops, particularly in the 1970s and 80s. There were major advances in some of these staple crops like rice and wheat. But where we need to be in terms of how productive our crops are and where we're projected to be if you look out to 2050 are quite far apart. So that's the dashed and the straight lines there. And so research that we do in model organism in plants is also essential to better understand what's going on in the food that we rely on. Okay. So the system we work in is Arabidopsis. So Arabidopsis thaliana, some of you, probably many of you have never heard of till Harvey introduced it a moment ago. It's actually the plant about which more is known than any other plant in the world. And it's a very, very modest organism. So its generic name is Thalecress. It's part of the mustard family. But in the 1990s, it really became this power horse for plant biology research due to the efforts actually of Jerry Fink at Whitehead and others in the field. And so why do we study this plant so intensively? Well, the reason it's such a good organism for biomedical research is it has many of the features that you would want in any organism that you were doing experiments on. So it has a short life cycle. for a plant. I mean, it's still a couple of months, but it has a relatively short life cycle. And that's great because that means the shorter the life cycle is, the more experiments we can do. It's very easy to grow, and it doesn't take up much space. And so these very practical considerations are important in science. We have a nice greenhouse up on the roof of the Whitehead, but if we had to grow, and we can grow thousands and thousands of Arabidopsis plants, but if we were growing thousands of rice plants or thousands of corn plants, it would just simply be impossible. Also, all of the molecular biology tools that you would want in an organism that we think of as being available in mice or fruit flies are available in Arabidopsis. So we have the ability to mutate genes now with CRISPR, to edit genes, and to add genes back to the plant. And so because we can manipulate genes in the plant, we can determine what their function is. Okay. So in my lab, we're interested in understanding, as David mentioned, epigenetic regulation of gene expression and how that affects plant growth and development. Now, many of you have probably heard this word epigenetic. Either in a scientific context or in the popular press. It's something that is bandied about quite frequently. But its meaning can be a little bit mysterious, even to those of us in the field. So I'll just start by defining this word for you and then telling you more about it, what it means and what exactly we're looking at in plants. Okay, so you're all familiar with the concept of genetics, that the sequence of genes, your DNA sequence, genes have a certain sequence, and that controls traits. And so here I'm showing you an example of so on the left that's a normal looking Arabidopsis plant. It's wild type, there's no mutations. So this is a plant that has started to flower. That long stalk there has flowers at the tip. And so we know a gene that controls when the plant decides to flower. It happens to be a gene called FWA, and in this plant it would have a particular sequence. Now, if that gene was mutated, if we changed a base, one of the DNA bases in that gene, what might result is a phenotype like this, where the gene is mutated, it's not active, and so the plant doesn't flower at the appropriate time. And what happens when a plant doesn't flower at the appropriate time is it makes more leaves. So here on the right is a late flowering plant. And so we would call this a genetic change. So there's a gene that's mutated in those plants on the right. So we define genetics as heritable changes in trait caused by changes in the DNA sequence. So what's epigenetics? So you know that the sequence of genes determines traits, but not all of the time. And so in fact, in this example here of these two plants that look very different, the difference is an epigenetic one. So people discovered these mutants. They knew the gene that controlled flowering, and they looked at the sequence of that gene, and it was absolutely identical between the plant on the left and the plant on the right. So what's going on? There's some epigenetic difference here. And so we define epigenetics as heritable changes in traits not caused by changes in the DNA sequence. So you can think of epi as meaning above, so things that are acting above the level of the DNA sequence. And these epigenetic differences have been described. There are many examples of these differences, particularly in plants, also in animals as well. And I'm giving you two classic examples here. So on the left are a type of European pear. And that pear is normally red. But sometimes on these plants that make red pears, you'll get green pears. And it's not because they're not ripening. It's just the skin remains green, even in a ripe pear. And so the gene that controls this trait, how red the skin of the pear gets, is known. You compare the sequence of the gene between this green one and the red one, and it's exactly the same. So we conclude that there must be some epigenetic difference between these two fruits. On the right here is a very famous example from mice, where you can see these mice have differences in coat color. The mice on the left are blonde. On the right, they get darker. And the gene that controls coat color is known. And again, in these mice, which are siblings, the gene has the exact same DNA sequence. So how can this be? What do we know about the molecular basis of epigenetic differences? In all of our labs at Whitehead, we're interested in understanding molecular mechanisms. And what's been discovered from research in plants and in animals, a lot of the discoveries actually were made in Rudolf Jaenisch's lab in his work with mice, are that the way the DNA is packaged in the cell and modified affects how it's interpreted by the cell. So all of us have genomes that if you stretch the DNA from end to end would be about 2 meters long. So maybe that's about this long. And that DNA has to fit inside a cell inside a nucleus that's about 10 microns in size. So that is 9 orders of magnitude difference. So you have to take something that's this big and fit it into something that is, we cannot see. And so the way that does that is the DNA is packaged. So I've listed a few things here. On the right is the familiar double helix of DNA. That DNA can be modified chemically, and I'll focus on that, which can affect its packaging. But DNA is wrapped around proteins. All the DNA in your cells are wrapped around proteins called nucleosomes. The DNA is negatively charged. The proteins are very positively charged. And so they attract one another. And that DNA that's wrapped around protein can then form higher order structures, the nature of which are still not understood, that ultimately lead to this very compact form that you find inside cells. And so the way the DNA exists in your cell, it's sort of, in addition to its sequence, how it is packaged or modified affects its function. Okay. So the modification that I'm going to focus on today is one called DNA methylation. So of the 4 bases in plant and animal DNA, A, T, C, and G, one of them, cytosine, or C, can be modified by enzymes that are called DNA methyltransferases. So on the left here is the structure of cytosine, the cytosine base. And that can be modified by DNA methyltransferase enzymes to generate something called 5-methylcytosine. And so you can see that the difference between these 2 structures is that there's now a CH3 added onto the cytosine on the right. So that's just a carbon with 3 hydrogens bonded to it. Very, very simple change. But that simple change has a very large impact on how the DNA is interpreted by the cell. So in general, when this base, this modified methylated cytosine, or as I'll refer to it, just DNA methylation, is found in the DNA, that DNA tends to be transcriptionally silent, as we call it, or not read by the cell. So the DNA is there. It may have a perfectly fine sequence, but it is not recognized by the machinery that would make RNA. So this cytosine methylation is found in a wide variety of organisms, from humans down other vertebrates, fish here, and plants. And so about 6% of our cytosines, of all of our bases are methylated. Similar fraction in fish and in plants. Plants actually have quite a wide variation in how much the DNA is methylated. So in Arabidopsis, it's about 5% of the DNA. In a genome like wheat, it's about 30% of the cytosines that are methylated. Now, interestingly, you'll note that so some of these organisms on the bottom here are our other favorite laboratory models. Fruit flies, this is the nematode C. elegans. Here on the left is the budding yeast. And so those are great systems for biological discovery, but they actually don't have DNA methylation in their genomes. They have other ways to control their genes. And so this is another reason that plants have been this great model system for discoveries in this field, because a lot of the other organisms that are really easy to work with, like fruit flies, simply don't have this modified base. So the plants are more similar to humans in this regard. Okay. So if we get back to these examples that I mentioned, what's going on here in both cases is in these organisms that show different traits, there's a difference in DNA methylation at the relevant gene. Okay. So in pairs, this gene, which I'll represent by a blue box, it's called MYB10. It's required for the production of the red pigment. So it's a gene that controls the expression of other genes. So that gene needs to be on in order for those pairs to accumulate that red pigment. So what's happening in the green pairs is that the regulatory sequences of that gene, which we draw as upstream or to the left of the gene, have become DNA methylated, which I'll indicate throughout the talk with these red lollipops. And so that methylation in those regulatory sequences actually shuts the gene down. So this gene is not active, indicated by the lack of the arrow, and the pairs stay green. Similarly, these mice, this gene that controls coat color, is on in the mice that make the brown hair follicles, but off in those with the blonde hair follicles. And that's because, again, there's methylation that is accumulated at that gene and turned the gene off. Now, the mice also illustrate something very interesting with regard to epigenetic phenomenon, is that you can see that some of them are a mix of blonde hairs and brown hairs. And if you looked at those individual hair follicles, there would be the blonde cells, the blonde hair cells would have the methylated copy, and the dark ones, the unmethylated copy. And so you can have these cells in the same organism in different epigenetic states. And so that's another feature of epigenetics, is that it's not as stable as genetic changes. A genetic change will vary. If you change the identity of a base, let's say a G to A, it's very rarely going to change back. But DNA methylation is not as stable. So methylation can be added, but can also be lost or even actively removed. And so these mice are actually a mosaic in terms of the epigenetic state of this gene. Okay. Now, DNA methylation is really, really important in our own biology, and in particular with regard to some diseases. And so one of the hallmarks of cancer that it's been known for quite some time is that cancer cells have a very different methylation profile. It's not that they've lost methylation or gained methylation, it's that the patterns are all wrong. So in typical cells or normal cells, genes don't have much methylation. There may be a lot of methylation in sequences that we don't want to express, things called transposable elements, which are selfish DNA. But what happens in cancer is you see everything shift. So repetitive DNA is losing methylation and may be active when it shouldn't be, and genes are gaining methylation, shutting them down. And so if this gene that gained methylation is one of these tumor suppressor genes that Bob Weinberg has identified, that can be very, very bad for the cell. Okay. So we're focused on this in plants. Why are we studying epigenetics in plants? Well, in fact, many of the discoveries that were made in this field were actually first made in plants. So the identity of the first DNA demethylase was actually from work I did as a graduate student, so that discovery was first made in plants. The other reason, which I've alluded to, is that the machinery involved in modulating where DNA methylation is added is very, very conserved between plants and animals, even though they are separated by about more than 1 billion years of evolution. And so one thing that's been important is that if we disrupt these genes that control methylation patterns, in animals, if you disrupt those genes, many of those mutations are lethal. But in plants, we find that we're able to mess with the epigenome a little bit more and still have a viable plant that we can work with. Okay. And then finally, more broadly, many of the agriculturally important traits, for example, when a plant decides to flower, are influenced by epigenetic regulation. Okay. So the reason I'm personally very interested in this field is because of the phenotype shown here. So these are seeds of this plant, Arabidopsis, and the seeds on the top are normal, typical seeds. Whereas the seeds on the bottom have a mutation that disrupts an enzyme that removes methylation from DNA. And so the consequence is that in these mutants, there's more methylation than there should be. And you can see that the result is not a good one. It's a little bit dark here on the screen, but those seeds, they look like raisins. They're very shriveled, and basically they're dead. So if you disrupt the pathway that is normally removing methylation in the plant, this is what you get. You don't make any viable seeds. Seeds, of course, as the next generation of plant are absolutely essential. They're also essential for us. So seeds worldwide account for probably 70% of the calories that are consumed. And this is primarily through rice, wheat, corn, and a few other grains. So on the left here is the seed we work on, Arabidopsis, but many of the findings we think are applicable or are known to be applicable to these other important plants. Coconuts I don't know about, but they're also a seed we eat. So nobody's researching coconuts. So why do those seeds with altered DNA methylation, they have more methylation than they should, why do they have that phenotype that's very, very severe where they die? Well, we know that this connects to a phenomenon called gene imprinting, or imprinted gene expression. And this is a phenomenon where genes are expressed depending on which parent they were inherited from. So each of us got one copy of every gene from our mother and one copy of every gene from our father, right? Same is true in plants. Plants have mothers and fathers. So one copy from mom, one copy from dad. Now, for most of those genes in our genome, both copies will be active. But for imprinted genes, something different is going on. Only the copy from the mother is active for a certain number of genes, or only the copy from the father. And this is truly an epigenetic phenomenon because you can have genes on chromosome from mom, on chromosome from dad. They have very, very similar sequence. They're in the same cell. One is active and one is not. And so this phenomenon of gene imprinting is basically found in only two classes of organisms, in mammals like us, in placental mammals, and in flowering plants. And I can talk to you over coffee if you want to know why that might be, that these very divergent organisms would be the only one that show this imprinted gene expression. And so there are a number of genes in the genome that we described as maternally expressed imprinted genes, or MEGs for short. So only the mom's copy is active, or paternally expressed imprinted gene, or PEGs, where only the copy from dad is active. And what we know is that differential DNA methylation is critical for establishing this difference in activity. So for example, here for this maternally expressed imprinted gene, what we often find is the paternal copy has DNA methylation and the maternal copy does not. And in fact, that underlies the reason that those seeds that I showed you with altered methylation are dead, because the imprinted genes imprinting is not being properly established. So this is also, so we study imprinting in plants. The other organisms it occurs in is placental mammals. And it's actually, there are a number of human diseases that are known to be associated with imprinting. So I thought I'd just tell you about one example, which is called Engelmann Syndrome, which is not incredibly rare and causes various developmental disabilities. And the reason this happens is because on our chromosome 15, there's a region that is imprinted. And in this region is a gene called the UBE3A. And the copy inherited from the mother is active, as indicated by the arrow, but the copy inherited from the father is not. So in individuals with this syndrome, what has been found is that they often have a deletion of that region inherited from the mother. And so the UBE3A gene is gone. It's just simply not there on the maternally inherited chromosome. There's a perfectly good copy on the chromosome from dad, but it doesn't matter because that copy is always silent, because the cells have decided only to express the maternal allele of this gene. And so if you had the reciprocal case where it was the dad that had a copy that was deleted, that would actually be fine, because it's only what's happening on the maternal chromosome that matters for a gene like this that's imprinted. So some of the things we've been doing in Arabidopsis are, first of all, using genomic approaches, as David alluded to understand what are all of the imprinted genes in a seed. And so we've done that now, and basically what we do is, this is just a cutaway of a seed and its component. It has an embryo. It has a placental-like tissue called the endosperm. And so we isolate these different tissues, and we do whole genome expression profiling. So basically we can, using some of the new high-throughput sequencing techniques that are available to us, at Whitehead we have a very nice genome core that works with us on this. We can see what are all the genes that are active in these tissues, and we can actually tell whether or not they're active from the maternally inherited copy of that gene or from the paternally inherited copy of that gene. And most genes are active from both copies. So this is some actual data here, looking at all the genes that are expressed and what percent of activity comes from the maternal copy. And so most have about a bit more than 50% from the maternal copy, but there are some genes over there where 100% of activity is from the maternal copy, or on the left, fewer genes where 100% of activity is from the paternal copy. And so we know the identity of these genes. Some of them are sort of classified as unknown genes. Some we have a good idea what they do. And they're involved in very, very fundamental cellular processes like controlling cell division, in the biosynthesis of hormones, and most interesting to us, genes that regulate the expression of other genes through some of these epigenetic mechanisms I told you about, like changing the packaging of the DNA. And so what we're finding when we look at imprinting is that many of the genes that are imprinted are themselves genes that we would expect to then modify the epigenome. And so we're very interested in understanding what are these networks of gene expression that are created through this process. Another thing that we've looked at is trying to understand how variable imprinting might be. And so in Arabidopsis is a species that's distributed throughout the world. It's in Europe, Asia, near the equator, in North America as well. And so on the left here are two different normal strains that you would find, in nature, strain 1 and strain 2. And what I'm showing you is if you take those two strains and cross them together, so you do a controlled mating, what do the seeds that are produced look like? So strain 1 up there in the upper left corner, if you take a female that's strain 1 and a male that's strain 1, you get those seeds in the upper left. They're, look pretty normal, a little bit small. Strain 2 in the bottom right, if you cross it with itself, you get quite large seeds. But the interesting thing is that if you cross these two together, you get seeds that differ in size depending on who was the mom and who was the dad. So here on the top right in the orange box, if we take strain 1 as the female and cross it to strain 2 as the male, we get seeds that are small, whereas if we take strain 2 as the female and strain 1 as the male in the green box, we get very large seeds, larger than just the strain 2 seeds on their own. So those are seeds that have the exact same parent. It just depended which one served as the male in the cross and which one served as the female. And so we've looked in crosses like this at imprinting, and we found that imprinting is variable from strain to strain. About 10% of the imprinted genes vary. And we've been able to connect this to naturally occurring differences in DNA methylation that exist in populations worldwide. So strain 1 is from Europe here, and strain 2 is from the Cape Verde Islands. And so now what we've been able to do, so we have a lot of correlations between the methylation status of a gene and its imprinting. Now we've been able to modify imprinting by changing the methylation status of genes. So we've been able to take a gene that wasn't imprinted in strain 2, add methylation to it, and make it now imprinted. And so this both tells us that methylation is causal for imprinting, but also that we may ultimately be able to engineer this system epigenetically. And so some of the questions that we're interested in going forward is what's, understanding this in much more molecular detail, what's the relationship between DNA methylation and some of these other epigenetic marks that I alluded to? how do you actually maintain epigenetic asymmetry through cell division? So there's this difference in methylation between the copy from the mom and the copy from the dad. But the methylation system is active in those cells. So why is mom's copy methylated and dad's copy isn't? So once you find an asymmetry, you're always then working back to the next asymmetry. How did you establish that? We're interested in understanding the functions of these genes during seed development, particularly those that look like they may be regulating other aspects of the epigenome. And then ultimately, a related question is whether or not we can modify imprinting to actually be able to do crosses that are currently impossible, either between different species or between plants of different ploidy. And actually, we have some evidence that modulating imprinting can actually increase the ability to cross plants of different ploidy. More broadly, what I'd like you, as you're telling your friends and family, what you learned today, what I'd like you to take away from this talk is that the way DNA is modified or packaged has a profound impact on its activity, that we can study this modification that I've told you about, DNA methylation, within plants, and because, of course, of the fundamental conservation of biological systems throughout evolution, what we find in plants we can apply to many other organisms, including ourselves, that you actually need to maintain normal patterns of DNA methylation throughout growth and development, throughout cell division. And when an organism does not do that, that can lead to developmental defects, as I showed you with the plant seeds, or to diseases like cancer. And finally, I hope I've convinced you, and Jinka will convince you more, that plants are really a fantastic system for studying these fundamental conserved mechanisms. So I'll just end by showing you the happy people in the lab who are involved in all of these experiments. So we're a mix of graduate students, postdocs, research technician, and a couple of MIT undergraduates, which are really a great group of people to work with, very enthusiastic, very passionate about what they're doing. And I'd also like to acknowledge my funding. So federal funding is, of course, critical. So all of you as taxpayers have paid for part of this work. So we have funds from the National Science Foundation and the National Institutes of Health. So thank you for supporting that. Some foundation grants, and of course, tremendous support from Whitehead Institute itself. Thank you.
Yeah. Are there questions for Mary?
Don't be shy.
Yeah, don't be shy.
Yeah.
Methylation.
Oh, it's all about
what?
context.
It's
so
context.
you want it in the right places. So I would say it's a good thing. You want to eat things that will provide folate, which is ultimately where methylation comes from chemically. So you want to have things in your diet that will give you plenty of methyl donors. Plants obviously make their own food, but methylation in the right place is absolutely essential. It's a good thing. It's when the system goes awry, where methylation is where it shouldn't be, or it isn't where it should be, that there are problems. Yeah. Yes?
If you have a cancer cell that's related to methylation, can you reverse that in the mature cell as the protein turns over, or is it only in the development of the new cell?
So that's a good question. There are actually some drugs that have developed that are methylation inhibitors. So a molecule called 5-azacitidine, it basically is a molecule that looks like cytosine, and so it replaces cytosine, and then it can't get methylated. So it's a methylation inhibitor, and actually that's been used with some success to treat some blood cancers, I believe. So being able to reverse that epigenetic state that arises in cancer is something that people are very much trying to do. Yeah. Yes?
So you started the conversation talking about the
So that is ultimately where this could be applied. So, like all the labs at Whitehead, we're interested in these fundamental cellular processes, but we are discovering genes that, when you modulate their expression, affects the final size of the seed, and affects the development of this tissue inside the seed called the endosperm, which is actually where our calories come from. And so the idea is that if you can understand these processes, you may be able to modulate them and get bigger seeds, which has been a goal of agricultural biotech companies for many years. There's been a lot of research trying to make big seeds. And it's clear that some of these genes that we're studying are going to be important in that process. Yeah, basically a big seed is the mother plant is putting more resources into that seed. So you want to switch the balance so that resources are going into the seed and not into making leaves or something. I guess we'll start in the back and move forward. So John.
So there's the concept of methylation. It applies to genes, yes, but does it also apply to non-coding DNA, such as centromeres and telomeres and so forth?
Absolutely. Very, very good question. So I didn't get into this. I just focused on genes because we all think about genes. But where methylation is really, really important, getting to your question, is in some of this non-coding DNA, like centromeres, like what's called transposable elements, which are selfish DNA that can jump around the genome and create havoc. And so the way that cells silence that DNA is through methylation. And so a major function of methylation is as a defense against this non-coding DNA in our genomes. Alan?
Yeah, my question is a follow-up on both of those other ones. Is you're saying you could manipulate the methylation to get more calories, which people need by 2050. But it also means there'll be different creations of proteins and different ratios of proteins. So my question is, would that lead to allergies? Would somebody form an adverse reaction to the different proteins or the combination of proteins?
It's certainly possible, just like you can have an allergy to anything that you eat. So the question is, if we're making a bigger seed, does it have the same components of the smaller seed, just more of them? Or is there some fundamental change? So we don't know that. We actually have started doing some, someone inspired by the things that Jinka does is looking at the metabolites in seeds, looking at the fat content in some of these different imprinted gene mutants. So, what I will say is that everything we eat is, not in a natural state. It's all been bred over 10,000 years of human farming. And so everything that we eat, may be modified in certain ways by different combinations of genes. And sometimes that may cause an allergy. Most of the time, probably not. Yeah.
This isn't really a scientific question. It's more maybe for David, along the lines that Whitehead looks for sources of support from what you might call the medical industrial complex, pharma companies, the deep pockets. Does the biological or the mechanical aspects of research at Whitehead draw the attention of the agri-farm industry, and are we looking there for?
Why don't you take it?
Well, I've certainly talked over my time at MIT. So MIT has this industrial liaison partnership program, and I have certainly talked with people at Monsanto through that, through European ag biotech companies. I haven't, gotten any funding from them, but this is certainly something that those companies are aware of and are interested in.
We have time for one last question.
So we do gene sequencing to identify mutants or variations to identify what the respective traits may be. Does that gene sequencing also identify the variations that are epigenetic, such as methylation, or do we need other screening techniques to understand the methylation of the gene sequence?
Yeah, that's a really great question. So the way we identify where DNA methylation is, and we can do it at single base resolution, right now the most commonly method used in the field is called bisulfite DNA sequencing, where basically if you take DNA and you treat it with this chemical sodium bisulfite, that reacts differently with methylcytosine and cytosine. And so then you sequence that DNA and you can tell where that DNA was methylated or not, whether that cytosine was methylated or not. So it's not direct sequencing of the DNA, but once you chemically treat it and then sequence, we can tell where methylation was. There are technologies being developed to try and directly detect these modified bases in a sequencing reaction. It can be done for methyladenine, which is something that you find in bacteria. Right now, it doesn't yet work for methylcytosine, but this is probably something in the future that we'll just be able to take the DNA and directly find both sequence and methylation status.
All right.
Oh, okay. Sorry. Yeah, go ahead.
Yeah.
Nutrients related to this higher yield. In other words, well, I think,
Yeah.
do we need more nutrients for the soil, for the environment of that seed,
Yeah.
in order to get that higher yield as well?
Yeah, I mean, of course, I've presented you a very simplified version, but all of these things come into play when you're talking about agriculture, what's in the soil, what's actually the nutritional quality of the seed itself. So if it's a bigger seed but is less nutrient rich, that may not be something that is worth going after. So you need to take an integrated approach to all of this.
Mary,
Okay.
thank you for a wonderful overview of your work. So we're going to take a 10-minute break now. You can refill your coffee cups, and let's be back in place in 10 minutes.